Hello, and welcome back, or perhaps for the very first time, to the Sleepy Wiki. Take a moment now to let your shoulders gently drop, release any lingering tension, and allow your breathing to slowly deepen, becoming soft and rhythmic. Tonight, our whispered exploration leads us to a concept both ancient and profoundly intricate: the gene. Imagine, if you will, the very blueprint of life itself, a delicate, almost mystical instruction passed down through countless ages. It is a subtle hum within every living thing, a secret code guiding the shape of a leaf, the color of a butterfly's wing, and even the quiet patterns within your own unique self. We'll gently trace these invisible threads of inheritance, these whispers from the deep past that continue to weave the tapestry of existence. If these quiet journeys bring you a sense of calm and gentle rest, you might consider softly tapping the like button, or perhaps even subscribing, to help more weary travelers find their way to our peaceful haven. But for now, let us begin our quiet contemplation of these fundamental, wondrous components of life itself. To understand these whispers of instruction, let's cast our minds back, far, far back in time. Imagine, if you will, our Earth as a much younger world. The oceans were warm, teeming with simple chemicals, a vast, soupy cradle we sometimes call the primordial soup. There were no complex creatures then, no trees, no animals, not even the simplest cells as we know them today. Yet, even then, the whisper of information, the earliest stirrings of instruction, began to emerge. In this ancient sea, tiny molecules were bumping into each other, forming fleeting connections. And among them, a remarkable molecule began to take center stage: RNA. Think of RNA as the enthusiastic, versatile apprentice of early life. Unlike its more famous cousin DNA, RNA had a dual talent. It could not only carry instructions like a tiny scroll, but it could also fold itself into intricate shapes, acting as a mini tool maker, speeding up chemical reactions, a role we now mostly associate with proteins. This period, where RNA likely performed both jobs, is often called the RNA world, a time when life's first stories were etched and acted out by the same molecule. But life, ever a marvel of innovation, found an even better way to safeguard its precious blueprints. Imagine trying to keep your most treasured family recipe on a delicate, single sheet of parchment. It's easily damaged, easily lost. RNA, for all its brilliance, was a bit like that, a single, somewhat fragile strand. So, over countless eons, a more robust solution evolved: DNA. Think of DNA as the wise, steady librarian, carefully cataloging and protecting the precious information within a strong double-stranded helix, a sort of molecular fortress that offered much greater stability and protection. The baton, or rather, the ancient scroll, was passed. RNA continued its vital work as a messenger and helper, but DNA became the master archive, the keeper of the grand library of life. Now, what were these first instructions, these earliest stories in life's library? They were, in essence, the very first genes. Each gene was a specific sequence within the DNA, or initially RNA, that held the code for building a particular protein. Imagine each protein as a tiny, intricate machine, a pump, a structural beam, an enzyme that performs a specific task. The genetic code, this fundamental language that translates gene sequences into protein building blocks, is almost universal across all known life. From the smallest bacterium to the largest whale, the same words mean the same parts. This profound universality is like a shared ancient language, a testament to our common ancestry, echoing back to those very first genes. But how did life go from a handful of basic stories to the vast, sprawling epic that is the tree of life today? One of the most powerful mechanisms is beautifully simple: gene duplication. Imagine you have a particularly useful story, a favorite recipe. If you make a copy of it, you now have two. One copy can continue its original role, ensuring life carries on as usual. But the other copy, it's now free. It can mutate, it can change, it can experiment without fear of immediately breaking something vital. Over vast stretches of time, these duplicated genes can diverge, gradually evolving new functions, new nuances to their stories. This is how entire families of genes arise, all descended from a common ancestor, each with its own special role, like different versions of a classic tale adapted for new audiences. Consider, for instance, the hemoglobin gene family, responsible for carrying oxygen in our blood. It's not just one gene, but a collection, each slightly different, optimized for different stages of development, one for the embryo, one for the fetus, one for the adult. These all trace their lineage back to ancient duplications, where a single ancestral gene was copied, and each copy then began its own evolutionary journey, refining its purpose. But gene duplication isn't the only way to write new chapters. Sometimes, life finds inspiration by sharing. This is where horizontal gene transfer comes in. Imagine entire paragraphs, or even full stories being traded directly between unrelated books, rather than being copied down through generations of the same book. This is particularly common among bacteria, where genes can jump directly from one organism to another, even across different species, often bestowing new abilities like antibiotic resistance or the capacity to metabolize new compounds. It's a rapid way for evolution to mix and match genetic material, like a bustling marketplace of genetic ideas, accelerating the pace of adaptation. And then there are the clever internal rearrangements. Sometimes parts of different genes can be cut and pasted together, forming entirely new genes with novel functions, a process called gene fusion. Or, conversely, a single long gene might split into two distinct genes, gene fission. It's like taking elements from different narratives and weaving them into a fresh plot, or splitting a meandering epic into a tighter duology, constantly remixing the genetic lexicon. And even seemingly random elements play a role. Imagine restless literary critics or wandering poets who move through the grand library of life, sometimes highlighting passages, sometimes inserting new verses. These are transposable elements, often called jumping genes, segments of DNA that can copy themselves and move to different positions within the genome. While some might be seen as disruptive, many have played a profound role in shaping genomes, sometimes even creating new genes, or altering how existing genes are regulated, adding new inflections to ancient stories. And most wondrous of all, sometimes new genes seem to arise from scratch, out of what was once considered non-coding DNA, the seemingly blank pages between the stories. This de novo gene creation is like a sudden, meaningful tale appearing from random scribbles, driven by subtle mutations that eventually create a sequence that can be transcribed into RNA and then translated into a functional protein. It reminds us that the potential for novelty is always present, even in the quietest corners of the genome. So, from the humble RNA of the primordial soup to the robust DNA library, from copying beloved tales to sharing them with neighbors, from remixing plots to finding new stories on blank pages, the evolutionary journey of genes is a continuous, dynamic narrative. It's a testament to life's boundless creativity, always building, always refining, ensuring that the grand story of existence continues to unfold in ever more intricate and wondrous ways. And as we consider these ancient origins, it's a beautiful thought to remember that many of these fundamental stories, these earliest genes, are still with us, deeply conserved across billions of years of evolution, quietly performing their essential tasks, connecting us all to that distant past. They are the bedrock upon which all the glorious complexity of life has been built. But how do these genes, once created and refined, actually do their work in the cells of living organisms today? Let's gently turn the page to explore that next wondrous chapter. Now, as twilight deepens around us, let us turn our gaze inward to the wondrous landscapes within us. We've spoken of the gene as a whisper of instruction, a tiny blueprint guiding the symphony of life. But what precisely is this whisper made of? How is such intricate information held within something so infinitesimally small? Imagine, if you will, shrinking down, not just to the size of a cell, but smaller still, until you can perceive the very molecules that dance and intertwine to form these profound instructions. At this scale, the gene reveals itself as a magnificent, almost architectural marvel. It is not a solid block of memory, but a delicate, yet incredibly strong ribbon of information, intricately woven from a special molecule we call DNA. Picture a long, ethereal ladder. But this isn't just any ladder. It's a ladder that has been gently twisted upon itself, perhaps like two vines gracefully coiling around a central axis, or a spiral staircase winding its way up into an invisible sky. This iconic shape, known as the double helix, is the very signature of DNA, and thus the foundational structure of every single gene. It's a design of breathtaking elegance, discovered by Watson and Crick, a testament to nature's genius in packing vast amounts of information into a stable and accessible form. Now let's look closer at the components of this spiraling ladder. The two long winding sides, or backbones, of our twisted staircase are built from repeating units of sugar and phosphate. Think of them as the sturdy, consistent banisters of our spiral staircase, providing unwavering support and structure. They are strong, resilient, and identical throughout their immense length, forming the unchanging frame upon which the magic unfolds. But it's in the rungs of this ladder where the true story the actual genetic message is written. Each rung is formed by a pair of what we call nitrogenous bases. There are 4 distinct kinds of these molecular letters, like an alphabet of life, and their names are adenine, thymine, guanine, and cytosine. For simplicity, we often just call them A, T, G, and C. These 4 letters aren't random. They don't just pair up with any other letter. No, they follow a beautiful, precise rule, like partners in an ancient, elegant dance. Adenine, A, always finds its partner in thymine, T. They fit together perfectly, like 2 pieces of a finely crafted puzzle. And guanine, G, always pairs with cytosine, C. This faithful pairing, A with T and G with C, is absolutely fundamental to how a gene works. It's the secret handshake that holds the 2 strands of the DNA ladder together across its middle. What holds these pairs together, these delicate rungs of information? It's not a strong, unbreakable bond, but rather softer, more transient attractions called hydrogen bonds. Imagine them like faint molecular magnets, gentle but persistent, allowing the A and T or G and C to cling to each other. These gentle bonds are crucial, for they allow the ladder to unzip temporarily when the information needs to be read or copied, much like opening a book to a specific page. It's a design that balances strength with flexibility, stability with accessibility. So, as you trace the path of one of the sugar-phosphate backbones, you'll find a sequence of these letters, A, T, G, C, following one after the other in an incredibly specific order. And on the other side of the ladder, faithfully paired, you'll find their complementary partners. If one side reads ATGC, the other side will, without fail, read TACG. This complementary nature is vital, ensuring that the genetic information is always preserved and can be accurately reproduced. And here's another subtle marvel. The 2 strands of our double helix run in opposite directions. We call this anti-parallel. Imagine 2 roads running side by side, but one is a northbound lane and the other a southbound. They're perfectly aligned, forming a single pathway, but their inherent directionality is opposite. This elegant arrangement is not just a quirk. It's a clever design that ensures the cellular machinery can read and process the genetic information correctly and efficiently. The real magic, the true blueprint, lies in the sequence of these paired letters along the length of our DNA ladder. Think of it as a vast, incredibly long book. Each gene isn't the entire book, but rather a specific, meaningful paragraph, or perhaps a chapter within that grand volume. This particular sequence of A's, T's, G's, and C's is the actual code, the instruction, the recipe for a specific trait or function. It's like a secret language, written in just 4 letters, but capable of conveying all the complexity and wonder of life. The order of these letters matters immensely, just as the order of letters in a sentence creates meaning. Cat is very different from echt, even though they use the same letters. In the same way, a specific sequence of these molecular letters spells out the instructions for building a protein, or regulating a cellular process, or indeed defining a characteristic that makes you uniquely you. So, a gene, in its molecular essence, is a segment of this vast, twisted DNA ladder, a specific stretch of those paired chemical letters, holding within their precise sequence a profound instruction. It's a marvel of miniature engineering, a testament to the power of information stored at the atomic scale. And as we understand how these instructions are physically stored, nestled within the graceful coils of DNA, our curiosity naturally turns to how this silent chemical script is brought to life, how it is read and translated into the active symphony of a living organism. But that, my friend, is a journey we shall embark upon as we softly transition from the quiet structure of the gene to the dynamic dance of its expression. With this understanding of structure settling into place, imagine with me now, if you will, that each gene we spoke of earlier isn't just a faint whisper of an instruction, but a meticulously penned tale, an intricate blueprint held within the grand library of our very being. But how is such a story written? How does a blueprint, nestled deep within the nucleus of a cell, become a bustling working part of us? The answer lies in a concept both profound and elegant: the genetic code and the delicate dance of information transfer. Picture this vast, ancient library filled with countless scrolls, our DNA. Each scroll holds countless stories, each story a gene, detailing how to build and maintain the incredible machinery that makes us, us. But these stories aren't written in a language we speak. They're composed in a secret universal alphabet, understood by almost all life on Earth, from the tiniest microbe to the tallest tree, and of course to each one of us. This secret alphabet has only 4 letters: A, T, C, and G. These stand for adenine, thymine, cytosine, and guanine, the fundamental building blocks or nucleotides of DNA. Think of them as tiny molecular beads strung together in long, magnificent chains. The magic truly begins when these letters are read not individually, but in groups of 3. These 3-letter words are called codons. Imagine these codons as precise instructions. For example, the codon GGC might tell the cell, "Bring me a glycine," while UAA might mean, "Stop, this story is finished." Yes, UAA, because when we switch from DNA's T to its close cousin, RNA, the T transforms into a U for uracil. It's like a slight dialect shift in our universal genetic language. Each of these 3-letter words, these codons, specifically codes for one of the 20 different types of amino acids, the fundamental building blocks that, when strung together, create proteins. It's a remarkably robust system too, sometimes called degenerate. This means that several different 3-letter words can sometimes spell out the same instruction, coding for the same amino acid. It's like having several synonyms for a word in a sentence, adding a layer of resilience to life's most precious instructions. Now, how does this beautiful coded blueprint transform into action? How does the quiet instruction in the library become a bustling activity in the cell? This is where the magnificent journey of information transfer begins. It's a 2-step ballet known as the central dogma of molecular biology, from DNA to RNA, and then from RNA to protein. Let's gently step into the first act, a process called transcription. Imagine a very special scribe, named rRNA polymerase, gliding along one of our precious DNA scrolls in the nucleus. This scribe's task isn't to copy the entire scroll, but only a specific story, a single gene. As the RNA polymerase moves, it carefully unzips a small section of the DNA double helix, revealing the ladder's inner rungs. Then, using one side of the DNA ladder as a template, it begins to build a new temporary scroll. This new scroll isn't made of DNA, but of a slightly different molecule called messenger RNA, or mRNA. Think of the mRNA as a temporary working copy of a specific recipe from the master cookbook. It's shorter, more portable, and perfectly designed to carry the message out of the nucleus and into the bustling cell cytoplasm. Just like the DNA, this mRNA molecule is built from the same 4 letters, but with uracil U replacing thymine T. So, where the DNA template might have a G, the mRNA will have a C. Where the DNA has an A, the mRNA will have a U. This careful complementary pairing ensures that the message is copied precisely, letter for letter, word for word. Once our mRNA messenger scroll is complete, it detaches from the DNA, and the DNA gently zips itself back up, safely preserving the original master blueprint. The mRNA, now carrying its vital instructions, embarks on a journey out of the nucleus, through tiny pores, and into the busy cytoplasm of the cell. This is where the second, equally wondrous act unfolds. Translation. Imagine the cytoplasm as a vast, microscopic city, and within it, busy little factories called ribosomes. These ribosomes are the master builders, the incredible molecular machines designed to read the mRNA message and assemble the specified proteins. Our mRNA messenger scroll arrives at one of these ribosome factories, threads itself through, and the process of reading the codons begins. But how do the ribosomes know which amino acid to bring for each 3-letter codon? This is where tiny specialized couriers called transfer RNA, or tRNA, come into play. Each tRNA molecule is like a dedicated delivery truck, designed to carry one specific type of amino acid. On one end, it holds its particular amino acid cargo, and on the other, it has a special 3-letter sequence called an anticodon. This anticodon is perfectly complementary to a specific codon on the mRNA. So, as the ribosome moves along the mRNA, reading each codon, the tRNAs with their matching anticodons arrive one after another, delivering their specific amino acid. The ribosome then acts like a skilled welder, forging a strong chemical bond between the incoming amino acid and the growing chain. One by one, amino acid after amino acid, the chain lengthens, forming a string of molecular beads, a polypeptide chain. This chain continues to grow until the ribosome encounters a stop codon on the mRNA, signaling the end of the protein's construction. Once released, this long, delicate polypeptide chain doesn't just remain a straight string. No, it immediately begins to fold and twist and coil into a precise, intricate, 3-dimensional shape, its functional form, It's like a dancer finding their pose or an origami master completing their creation. This incredibly specific shape is what allows the protein to perform its particular job. Whether it's building structures, catalyzing reactions, defending against invaders, or sending signals throughout our bodies. Every single function, every aspect of who we are, is fundamentally dependent on these perfectly crafted proteins. And so, from the silent, coded instructions of our DNA through the faithful messenger of mRNA and assembled with meticulous precision by the ribosome and tRNA, life's intricate machinery is built. It's a breathtaking symphony playing out billions of times a second in every cell within you, a testament to the elegant complexity of life. Each protein, a small miracle working tirelessly, all initiated by that gentle whisper of information flowing from the gene. Now, as these thoughts gently settle in your mind, let's prepare to softly explore another fascinating aspect of how these instructions are not only read, but also regulated, ensuring every story is told at just the right time and in just the right way. With the genetic code now unfolded before us, we've marveled at the blueprint itself, that incredible double helix of DNA, a vast library containing the complete instructions for life. But what good is a library, no matter how magnificent, if its precious books are never opened, never read, never brought to life? This, my dear listeners, is where the magic truly begins, with a process called gene expression. Imagine, if you will, that your entire being is a sprawling, bustling city full of intricate structures, tireless workers, and complex systems. Within each cell lies its master archive, the central vault where all the original, irreplaceable blueprints and recipes are safely stored. You wouldn't want to take these irreplaceable originals out onto the noisy factory floor, would you? No. That would be far too risky. Instead, the cell has a remarkably elegant solution for using these precious instructions to build and maintain its magnificent structures. It makes temporary, working copies. This journey, from the silent blueprint of a gene to a bustling, functional component of life, is the heart of gene expression, often beautifully summarized as the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. Let's gently follow one such instruction, a single gene, as it awakens. Our journey begins in the quiet sanctity of the nucleus, that protected inner chamber of the cell where the DNA resides. Here, a specific gene, a segment of that long DNA helix, contains the precise recipe for a particular protein, perhaps an enzyme to help digest your food, or a structural protein to give strength to your hair, or an antibody to protect you from illness. To access this recipe, a special molecular scribe, an enzyme called RNA polymerase, arrives on the scene. Picture this RNA polymerase as a diligent, meticulous librarian, carefully opening one of the great DNA books. It gently unzips a small section of the DNA double helix, momentarily separating its two intertwined strands. Like reading a secret code, the RNA polymerase then moves along one of these exposed DNA strands, the template strand, faithfully reading its sequence of bases, A, T, C, G. As it reads, it doesn't just copy. No, it builds a brand new, complementary strand, but not of DNA. This new strand is made of rRNA, ribonucleic acid. There's a subtle difference here, like a temporary note written in a slightly different ink. Where DNA uses thymine, T, RNA uses uracil, U. So for every A on the DNA template, the RNA polymerase adds a U. For every T, an A. For every C, a G. And for every G, a C. This newly formed RNA molecule is called messenger RNA, or mRNA, for it carries the message from the DNA. It's like a fleeting whisper, a single-use working copy of the precious recipe perfectly suited for the journey ahead. Once the RNA polymerase has transcribed the entire gene, the DNA strands zip back together, the master blueprint safe and sound, and the mRNA molecule detaches, now ready for the next stage. But wait, for eukaryotic cells like ours, this freshly made mRNA, often called pre-mRNA, isn't quite ready to leave the nucleus. It's a bit like a rough draft of a story, filled with useful information, but also some unnecessary bits. These unnecessary bits are called introns, sequences that don't code for the final protein. The valuable protein-coding parts are called exons. Think of introns as commercial breaks or tangents in a story, and exons as the crucial plot points. A brilliant molecular editor then steps in, performing a precise process called splicing. Like a skilled surgeon, this editor meticulously cuts out all the introns and elegantly stitches the exons back together in their correct order. It's an astounding feat of molecular precision, ensuring only the essential parts of the recipe remain. To further prepare this refined message for its perilous journey out of the nucleus and into the bustling cytoplasm, a protective cap is added to one end, the 5 cap, and a long tail of adenine bases, the poly-A tail, is added to the other. These additions are like putting a sturdy envelope around our precious message and adding a clear address label, ensuring its stability and helping it to navigate the crowded cellular environment. Now, and only now, is our mature mRNA ready to leave the sanctuary of the nucleus. Now our mRNA, now a polished and ready instruction scroll, exits the nucleus through tiny pores and enters the cytoplasm, the vibrant open factory floor of the cell. Here, a bustling assembly line awaits, the wall ribosome. Imagine the ribosome as a sophisticated molecular machine made of 2 main subunits, like 2 halves of a clamshell, that come together around the mRNA. It's the ultimate protein-building factory. The ribosome's job is to read the mRNA message and translate it into a chain of amino acids, the building blocks of proteins. But how does it read? The mRNA message is written in a language of codons, sequences of 3 nucleotide bases. Each codon specifies a particular amino acid. For example, the codon AUG is the universal start signal and codes for the amino acid methionine. Other codons specify other amino acids, and a few special codons act as stop signals. To bring the correct amino acids to the ribosome at the right time, we have another remarkable molecule, transfer RNA, or tRNA. Think of tRNAs as tiny specialized delivery trucks. Each tRNA molecule carries a specific amino acid on one end, and on the other end it has a 3-base sequence called an anticodon, which is complementary to an mRNA codon. So, if the mRNA codon is CGA, the tRNA with the GCU anticodon will arrive, carrying its specific amino acid. And LUN. The translation process unfolds in 3 beautiful steps: initiation, elongation, and termination. During initiation, the small ribosomal subunit attaches to the mRNA, usually near the start codon, AUG. The very first tRNA carrying methionine docks onto this start codon, and then the large ribosomal subunit joins, encapsulating the mRNA and the first tRNA. The assembly line is now complete and ready to roll. Next comes elongation, the heart of protein synthesis. The ribosome glides along the mRNA, reading one codon at a time. As it moves, a new tRNA carrying its specific amino acid arrives, its anticodon perfectly matching the mRNA codon currently in the ribosome. The ribosome then catalyzes the formation of a peptide bond between the amino acid brought by the new tRNA and the growing chain of amino acids already attached to the previous tRNA. The first tRNA, now empty, detaches and floats away to pick up another amino acid, ready to be recycled. This process repeats, codon after codon, like stringing beads onto a necklace, meticulously adding one amino acid after another, building a long linear chain known as a polypeptide. Finally, when the ribosome encounters a stop codon on the mRNA, like UAA, UAG, or UGA, the process reaches termination. There are no tRNAs that match these stop codons. Instead, special proteins called release factors bind to the stop codon, signaling the end of the protein-building process. The polypeptide chain is then released from the ribosome, and the ribosome itself disassembles, separating into its 2 subunits, ready to begin translating another mRNA molecule. Now our newly synthesized polypeptide chain is a long folded string of amino acids, but it's not quite a functional protein yet. Like a piece of origami that needs to be perfectly folded to take its final shape, this polypeptide must fold into a precise, intricate 3-dimensional structure. This specific 3D shape is absolutely crucial for the protein to perform its particular job, whether it's acting as an enzyme to speed up a chemical reaction, or as a structural component, or as a signaling molecule. Sometimes proteins even undergo further modifications, like having sugars or phosphates added, or being cut into smaller active pieces. They are then transported to their specific destinations within the cell, or even secreted outside, ready to perform their vital roles in the grand orchestra of life. And so, my dear listeners, this incredible molecular ballet of gene expression and protein synthesis unfolds within us every single second, millions of times over. It is the fundamental process that brings the genetic blueprint to life, building every enzyme, every structural component, every defense molecule that makes you, well, you. It's how a single cell can grow into a complex organism, how wounds heal, and how your body constantly adapts and responds to the world around you. It's a testament to the elegant, tireless ingenuity of life itself. As these intricate molecular ballets unfold within us, moment after moment, guiding the very essence of who we are, we begin to appreciate the profound elegance of life's instruction manual. But how exactly are these instructions passed from one generation to the next, ensuring that this incredible symphony continues? Perhaps that's a thought for another quiet moment. Yet knowing how proteins are built is only part of the story. Let us now explore the intricate world within each of your cells, a world far more complex and coordinated than any bustling city. We've talked about genes as the blueprints, the instructions for life. But what good is a magnificent library of blueprints if you don't know which ones to use or when to use them? Imagine if every instrument in a grand symphony played all the time, at full volume. It would be a chaotic noise, not a beautiful melody. This is where the magic of regulatory mechanisms comes in. The quiet conductors, the subtle lighting designers, the attentive stage managers of the cellular world. They ensure that each gene, each tiny instruction, is expressed at precisely the right time, in the right place, and in just the right amount. It's how a single fertilized egg can grow into a complex being with brain cells, muscle cells, and skin cells, all containing the exact same set of genes, yet performing vastly different roles. Each cell type is like a unique orchestra playing a different piece, using only the instruments it needs for its particular performance. Our journey into gene regulation begins even before the gene story is read, right at the very packaging of our DNA. Think of your DNA not just as a long, delicate thread, but as an incredibly long scroll, meticulously wound around tiny protein spools called histones. These spools, bundled together, form structures called nucleosomes, and these nucleosomes can be packed together very tightly or kept a little looser. When the DNA is tightly wound and coiled, like a scroll tied securely with many ribbons, it's very difficult for the cellular machinery, the readers of the gene stories, to access that part of the DNA. We call these inaccessible regions heterochromatin, quiet zones where genes are largely silent. But when these spools are loosened and the DNA unwinds a little, like a scroll gently unrolled, suddenly the instructions become accessible. These active, accessible regions are called euchromatin. It's like the stage lights are being turned on, ready for the performance. How does a cell decide to tighten or loosen these ribbons? Well, it's a fascinating molecular dance involving tiny chemical tags, not changing the DNA sequence itself, but adding small notes or flags to the DNA or its histone spools. This is the realm of epigenetics, a wondrous layer of control that sits above the genetic code. For instance, tiny chemical groups called methyl groups can attach to the DNA itself, often acting like a silent button, gently pressing a gene into slumber. Other tags, like acetyl groups, can attach to the histone spools, often acting like a wake-up call, prompting the DNA to loosen its grip and become ready for transcription. These epigenetic notes are remarkable because they can be influenced by our environment, our diet, even our experiences, and sometimes they can even be passed down from parent to child without ever altering the fundamental sequence of the DNA letters. It's like leaving annotations in the margin of a score, guiding the musician without changing the notes themselves. Once the DNA is accessible, the real work of transcription can begin, the process of copying a gene's blueprint into a messenger RNA molecule. Here, the cellular orchestra's conductor takes center stage. These conductors are special proteins called transcription factors. Imagine them as highly skilled molecular musicians and stagehands who know exactly where to stand on the DNA stage to either beckon the RNA polymerase, the enzyme that reads the DNA and builds the mRNA copy, or to politely block its path. They bind to specific regions of the DNA like a promoter region, which is essentially the gene's starting line, telling the RNA polymerase where to begin. Sometimes they bind to enhancer regions, which are like volume knobs, turning up the gene's expression, ensuring a strong, clear message is produced. Other times, repressors bind, acting like a mute button, dampening the signal or silencing it altogether. Think of it as choosing which story to read from a vast library. The transcription factors are the librarians who pick out the right book, place it on the reading stand, and even adjust the lighting so the reader, RNA polymerase, can see the words clearly. Without the right transcription factors, even an accessible gene will remain unread, its story untold. This precise control allows a brain cell to express genes for neurotransmitters, while a skin cell expresses genes for keratin, both from the same genetic library. But the story doesn't end with the creation of the messenger RNA, or mRNA. Our mRNA, fresh from the DNA library, often undergoes further editing and refinement before it's ready to deliver its message to the protein-making machinery. This is post-transcriptional control. Imagine our mRNA as a rough draft of a play script. Before it reaches the actors, it needs to be perfected. In eukaryotes, like us, our genes often contain sequences called introns, sections that don't code for protein, interspersed with exons, the actual coding parts. Special cellular editors come along and splice out the introns, leaving only the exons to be stitched together. This splicing is incredibly powerful, as sometimes a cell can even choose which exons to include or exclude, creating different versions of a protein from the same gene. It's like having a master chef taking a single recipe, and by choosing different ingredients or preparation methods, creating several distinct, delicious dishes. And then there are the quiet whispers of the microRNAs, or miRNAs. These are tiny, non-coding RNA molecules, like vigilant molecular detectives. They don't carry protein instructions themselves, but they patrol the cell, looking for specific mRNA messages. When a microRNA finds its target, mRNA, it can bind to it, essentially marking it for degradation, telling the cell to discard the message, or inhibiting its translation, preventing the protein from being made. It's a subtle yet potent way to fine-tune gene expression, like a silent alarm system that prevents unwanted messages from reaching their final destination. Finally, even after the mRNA message is refined and ready, the cell has control over translation, the actual process of building a protein. It can decide how efficiently that message is read by the ribosomes, the cellular factories that assemble proteins. And once a protein is made, it's still not the end of the story. Post-translational control involves modifying the protein itself. Tiny chemical tags like phosphate groups can be added or removed, acting like molecular on-off switches or adjusting the protein's activity, much like adding an ingredient to a finished dish to change its flavor. And when a protein has served its purpose, or if it's faulty, the cell has an elegant recycling system, the ubiquitin proteasome system, which tags unwanted proteins with a ubiquitin marker, signaling them for gentle breakdown and recycling, ensuring cellular tidiness and efficiency. So, you see, the journey from a silent gene to a working protein is not a simple straight line, but a beautifully choreographed ballet. Each step, from the packaging of DNA to the final fate of a protein, is precisely controlled by a vast network of regulatory mechanisms. They ensure that your body's cells are not just a cacophony of constant activity, but a symphony of life, playing a harmonious melody with incredible precision and grace. This exquisite control is what allows life to be so diverse, adaptable, and wonderfully complex. And it's what ultimately shapes the traits and characteristics that make each living thing unique. Now, as we appreciate this silent choreography that unfolds within you, let's gently consider how these intricate mechanisms come to be and how these coded instructions are passed down through generations, shaping the tapestry of life itself. To pass down these instructions through the ages requires a feat of extraordinary organization. Imagine, if you will, the longest, most delicate thread you could possibly conceive. A thread so infinitesimally fine that if you were to unspool just one of these threads from a single human cell, it would stretch out for about 2 meters. 2 whole meters of incredibly detailed instructions, all wound up and tucked away inside a microscopic chamber, smaller than the period at the end of this sentence. This thread, of course, is our DNA, and that tiny chamber is the cell's nucleus. The challenge, you see, isn't just containing this immense length, but organizing it so that every piece of information, every gene, can be found and read precisely when needed without getting tangled or lost. It's like trying to fit an entire library, filled with countless stories, into a thimble, yet still being able to pull out just the right book, open it to the correct chapter, and read the exact paragraph you need, all in an instant. This wondrous feat of biological engineering is what we call genomic organization. Now, our DNA doesn't just haphazardly coil into a ball. Oh no, it's far more sophisticated than that. It undergoes an astonishing process of compaction, a masterclass in elegant storage. The first step involves a special set of proteins, little molecular spools called histones. Think of these histones as tiny, perfectly shaped bobbins. Our long DNA thread wraps itself neatly around these histone spools, about 1.65 times, forming what we call a nucleosome. Imagine miniature pearls strung along a delicate necklace. That's what nucleosomes look like, beads on a DNA string. Each pearl is a nucleosome, a package of DNA wrapped around histones, making the DNA roughly 7 times shorter than its extended form. But the journey into compactness doesn't end there. These nucleosome pearls then begin to coil further upon themselves, forming a thicker, more intricate, rope-like structure. This rope then folds and loops into an even more compact form, a sort of supercoil. All these layers of coiling and folding create what we refer to as chromatin. Chromatin is the sophisticated material that makes up our chromosomes, those X-shaped structures we often see in diagrams. So, from a 2-meter strand, we arrive at a structure so tiny it can fit within the nucleus. Yet every gene retains its precise location. Now, here's where it gets truly fascinating. Not all parts of this meticulously folded chromatin are equally accessible. Some regions remain loosely packed, like an open book, easy for the cell's reading machinery to access and interpret. These active, gene-rich areas are called euchromatin. They are bustling with activity, like a vibrant marketplace, where genetic messages are constantly being exchanged and put into action. Here, the genes are often found, ready to be transcribed into RNA and ultimately translated into proteins, building blocks and workers of life. Conversely, other regions of the chromatin are much more tightly wound, deeply condensed, and largely inaccessible. These dense, often gene-poor areas are known as heterochromatin. Think of them as ancient, sealed scrolls in a forgotten archive. Information is there, but it's locked away, usually silent, and not actively read. This difference in packaging, from open euchromatin to tightly wound heterochromatin, is a crucial part of how a cell regulates which genes are active and which are not. It's like having a master switch for entire regions of the genome, controlling vast landscapes of genetic information. Within this organized landscape lie our genes. But even a gene isn't just one continuous, unbroken story. Especially in complex organisms like ourselves, genes are often like stories interspersed with riddles or blank pages. These coding segments, the parts that actually carry instructions for making proteins, are called exons. Imagine them as the meaningful sentences and paragraphs of a book. However, nestled between these exons are non-coding segments known as introns. These introns are like commercial breaks in a television show, or perhaps decorative borders or blank pages between the story chapters. They are part of the original script, but don't carry direct protein-making instructions. Before a gene's message can be fully utilized, these introns must be carefully snipped out and the exons precisely stitched back together in a process called splicing. It's an intricate editing job, ensuring only the truly relevant genetic information proceeds to the next stage. And how does the cell know which genes to read and when? This is where the surrounding neighborhood of a gene comes into play. Genes don't exist in isolation. They are surrounded by a symphony of regulatory sequences. Imagine tiny signposts and control panels on the DNA itself. There are promoters, which are like the "Start Here" buttons, sequences that signal where the gene begins. Then there are enhancers, distant control switches that can dramatically boost a gene's activity, even if they're located far away on the DNA thread. Conversely, silencers can dampen or turn off gene expression. These regulatory elements are the orchestra conductors of the genome, dictating the tempo, volume, and timing of each gene's performance. Their precise location and interaction within the three-dimensional space of the nucleus are critical for proper gene expression and cell function. Interestingly, the genes themselves, with all their exons and introns and regulatory neighbors, make up only a surprisingly small fraction of our entire DNA. The vast majority of our genome, upwards of 98% in humans, is composed of what we used to call junk DNA. However, this term is increasingly being reconsidered as we uncover the subtle yet profound roles of these non-coding regions. Much of this non-coding DNA consists of repetitive sequences. These are stretches of DNA patterns that are copied over and over again, sometimes thousands or even millions of times. Some are short, simple repeats called tandem repeats, like little molecular echoes that happen one after another. Others are longer, more complex sequences that are interspersed throughout the genome, acting almost like molecular nomads, capable of moving around the DNA. These are often remnants of ancient viral infections or jumping genes, called transposons. While many repetitive sequences don't directly code for proteins, they play crucial roles in maintaining the structural integrity of chromosomes, in gene regulation, and in shaping the evolution of species. Some might be like the ancient runes on a scroll, not part of the active story, but holding clues to the scroll's history and stability. Others might serve as spacers, ensuring proper distancing between genes, or even as platforms for complex regulatory interactions. The genome, therefore, isn't just a simple linear script. It's a dynamic, multi-layered document where genes are carefully embedded within a rich and complex landscape of structural elements and regulatory instructions. This intricate dance of DNA packaging, gene structuring with introns and exons, and the sophisticated network of regulatory signals all contribute to the marvel of genomic organization. It ensures that life's instructions are not only stored securely, but are also precisely accessible and expressible, guiding the development and function of every cell in our bodies. As we consider this breathtaking level of order, this miniature universe meticulously arranged within each tiny nucleus, it makes us wonder even more about the nature of these individual genes themselves and how their specific sequences translate into the incredible diversity of life we see around us. Perhaps we can explore that wonder next. This elegant organization, however, is only the beginning of the story. We now arrive at a truly fascinating frontier, a layer of control that adds exquisite nuance to the grand symphony of our genes. Imagine, if you will, that our DNA isn't just a static instruction book, but a living, breathing script that can be annotated, highlighted, and even edited without changing a single word of the original text. This remarkable layer of control is what scientists call epigenetics. The word itself, epi, means above or on top of, suggesting something that sits on top of our genetic sequence, influencing how it's read and expressed. Think of your genes as all the musical instruments in a vast orchestra. Each instrument, a violin, a flute, a drum, represents a specific gene, holding the potential to play its part. Now, epigenetics is like the conductor of this magnificent orchestra. The conductor doesn't change the instruments themselves, nor does he rewrite the musical score. Instead, he decides which instruments play, when they play, how loudly they play, and even in what sequence. He brings certain sections to the forefront, quiets others, and ensures that the entire performance creates a harmonious, coordinated piece. This is the essence of epigenetic control. It dictates the expression of our genes without altering the underlying DNA sequence. Now, our bodies are comprised of countless different cells. The steadfast cells of your skin, the bustling cells of your muscles, the thoughtful cells of your brain, and the tireless cells of your heart. And yet, almost every single one of these diverse cells contains the exact same set of DNA instructions. How then do they become so wonderfully different? This is where the epigenetic conductor truly shines. It's the epigenetic marks that tell a nascent cell, "You, little cell, you will become a neuron." Silence all those genes for muscle proteins and activate those for transmitting electrical signals. And to another, "You, my friend, will be a skin cell. You need to activate genes for protective barriers and turn down everything else." It's like having the full blueprint for an entire city in every construction worker's hand. But each worker only builds their designated part, the library, the park, the school, guided by invisible temporary annotations on their personal copy of the plan. Now, let's gently explore some of these exquisite annotations. Two of the most common and profoundly impactful epigenetic modifications are DNA methylation and histone modification. Imagine your DNA as an incredibly long, delicate thread, spun tightly around tiny protein spools. These spools are called histones. If the thread, DNA, is wound very tightly around its spool, it's difficult for the cellular machinery to read the instructions on that part of the thread. It's like having a scroll so tightly coiled you can't unroll it to read the words. Conversely, if the thread is loosely wound, the instructions are easily accessible. Histone modifications are like little chemical tags that attach to these spools. Some tags act like gentle lubricants, loosening the grip of the spool and making the DNA more accessible, essentially turning a gene on. Other tags act like tiny clamps, tightening the grip, making the DNA less accessible and effectively turning a gene off. It's a subtle dance of accessibility, constantly adjusting which parts of the genetic script are open for reading. Then there is DNA methylation, a process that truly highlights the delicate precision of epigenetic control. Picture a specific letter in your DNA alphabet, a C for cytosine. Sometimes, a tiny chemical group, a methyl group, can be added directly to this C within particular regions of your genes, especially where many C's and G's, guanines, are clustered together. Think of it like placing a tiny, invisible sticky note over a specific word in your instruction book. This sticky note doesn't change the word itself, but it can subtly block the reader's finger from pointing at it or obscure it just enough that the reading machinery overlooks that section. When genes are methylated in these specific regulatory regions, it often acts as a signal to silence them, to keep them quiet and unexpressed. It's like a dimmer switch, turning down the light of that particular gene, or even turning it off completely. The most extraordinary aspect of epigenetics is its dynamic nature, its exquisite responsiveness to the world around us. Unlike the fixed sequence of our DNA, which remains largely unchanged throughout our lives, our epigenetic marks are remarkably plastic. They are influenced, sculpted, and reshaped by our experiences, our environment, and even our lifestyle choices. Imagine the delicate tapestry of your epigenome constantly being rewoven by the threads of your daily life. Consider, for instance, the profound impact of a diet. The foods we eat are not just fuel. They contain molecules that can directly influence the addition or removal of these epigenetic marks. A diet rich in certain nutrients, or conversely one lacking in them, can alter the methylation patterns on our DNA or the modifications on our histones, subtly shifting which genes are active or silent. It's as if the conductor adjusts the orchestra's performance based on the weather outside. A warmer, sunnier day encouraging a brighter, more playful melody, while a stormy one calls for something more subdued. And even more profoundly, stress and early life experiences can leave indelible epigenetic imprints. Studies have shown that challenging or nurturing environments in early development can shape an individual's epigenetic landscape, influencing their stress response, their mood, and even their susceptibility to certain conditions later in life. It's as though our early experiences write a kind of memory onto our genes, not by changing the fundamental story, but by adding annotations that suggest, read this part with caution, or emphasize this passage with hope. This fascinating interplay helps explain why identical twins, despite sharing the exact same DNA, can grow to be quite distinct individuals with varying health profiles and personalities, as their unique life journeys sculpt their epigenomes differently. And while the precise mechanisms are still being unveiled, there's even evidence to suggest that some epigenetic modifications can, under certain circumstances, be passed down through generations, not as changes to the DNA sequence itself, but as a kind of inherited memory of past environmental exposures or experiences. It's a whisper from ancestors, a subtle suggestion to their descendants about how best to interpret the genetic score in a world that might hold similar challenges. So, epigenetics offers a breathtaking vision of fluidity and adaptability, showing us that our genetic destiny is not entirely fixed. It's a dance between our innate script and the ongoing symphony of life's experiences. Our genes are the blueprint, but epigenetics is the ongoing construction and renovation, always responsive, always learning. It means there's hope and agency in how we live, knowing that our choices and environments can gently nudge the expression of our genetic potential. As we quietly ponder this intricate dance of genetic control, we can begin to appreciate the layers of wisdom embedded within our very cells. And as we consider the profound adaptability of our biological story, perhaps we can next turn our thoughts to the very beginning of this incredible journey. The moment a new life begins, and how our genes orchestrate the earliest chapters of development. Yet these responsive, adaptable instructions are not unchanging. Imagine now these intricate threads of DNA not just as static blueprints but as living stories, constantly being rewritten and reread within every single cell. Each gene, a precise paragraph in the vast library of life, holds instructions for building something vital, a protein perhaps, or a tiny regulator, guiding the symphony of your being. But even in the most carefully penned stories, sometimes a small change can occur. Think of it like this. Picture a long, ancient scroll filled with the delicate script of life's instructions. Most of the time, when this scroll is copied from one generation of cells to the next, it's done with exquisite fidelity, a perfect replica. But occasionally, just sometimes, a tiny ink blot might appear, or a letter might be accidentally transcribed differently, or even a whole word might be skipped or duplicated. These little alterations in the genetic script, these quiet typographic errors, are what we call gene mutations. It's important to understand that a mutation isn't always a dramatic event, like a page being torn from the scroll. Often, it's far more subtle. A single letter in the endless genetic code, adenine, guanine, cytosine, or thymine, simply swapping places with another. This microscopic shift might seem insignificant, but it's a profound spark. How do these tiny alterations happen? Sometimes it's a simple copying error, a slip of the cellular hand during the immense task of replicating DNA. Our cells are remarkably good at proofreading, but occasionally a mistake slips through. Other times, external whispers from the world around us, like certain forms of radiation or chemicals, can gently nudge these changes into being, leaving their mark on the delicate strands of our genetic story. Now, you might wonder, are these mutations always bad? The answer is a soft, reassuring no. Most often, these little changes are like a typo in a very long novel. They don't change the plot at all. They might occur in a section of the DNA that doesn't code for anything, or they might subtly alter a protein in a way that doesn't affect its function. We call these neutral mutations. They're like different shades of the same color, adding variety without changing the fundamental hue. Sometimes, yes, a mutation can be like changing a critical word in a recipe, causing the whole dish to turn out wrong, or even to become harmful. These are the mutations that can sometimes lead to genetic conditions, where a vital protein doesn't work as it should, gently disrupting the body's delicate balance. But then, in rare whispered instances, a mutation can be like an accidental tweak to an old recipe that makes it even better, more resilient, or uniquely adapted to a new flavor. These are the beneficial mutations, and they are the quiet engines of change, of progress, and of life's endless ingenuity. These myriad changes, these countless typos accumulated over vast stretches of time, are the very foundation of a natural variation. Look around, even in your mind's eye, at the tapestry of life. No two leaves on a tree are perfectly identical, no two snowflakes exactly alike. And within any family, siblings share so much, yet are wonderfully distinct. This beautiful, inherent difference among individuals within a species, that's natural variation. It's the rich palette of possibilities that mutations continuously paint, generation after generation. Imagine a forest full of ancient trees. While they are all trees of the same kind, some might have slightly thicker bark, making them more resistant to a particular insect. Others might have roots that spread a little wider, helping them find water in dry soil. Still others might produce seeds that can travel a bit further on the wind. These subtle differences, these variations, are all rooted in the genetic code, often born from those tiny mutations we spoke of. They are the unique gifts passed down through the generations, slowly accumulating and rearranging. But what determines which of these variations persist through time? Picture a gentle, invisible current flowing through this forest. This current is calm, natural selection. It's not a harsh judge, but more like a quiet librarian, subtly shelving the books of life. If the climate becomes colder, perhaps the trees with slightly thicker bark, or a metabolism that tolerates frost better, thanks to some ancient beneficial mutation, will be a little more likely to thrive, to survive long enough to have their own saplings. Their books, their unique genes, are thus more likely to be copied and passed on to the next generation. The trees with thinner bark or less frost tolerance might still exist, but their numbers might slowly, gently diminish over many, many generations. Over vast stretches of time, this gentle current of natural selection, acting on the raw material of natural variation, which remember comes from mutations, can lead to astonishing transformations. It's not a rapid, sudden leap, but a slow, persistent drift, like a river carving its path through stone, one grain at a time. This is how different species gradually adapt to their unique environments, how they change and diversify, eventually becoming something entirely new, yet always connected to their ancestral stories. The variations that offer even a slight advantage, a subtly better camouflage, a slightly more efficient way to digest food, a fractional increase in disease resistance, are gently encouraged to proliferate, to become more common within the population. So, as you drift toward sleep, let the thought settle that every unique shade of fur on an animal, every different pattern on a butterfly's wing, every slight variation in our own human family, from eye color to height to how quickly we heal, is a testament to this constant, subtle dance of change. It's a reminder that life is never static, but a ceaseless river of adaptation fueled by tiny, accidental genetic whispers. These whispers, these mutations, create the vibrant tapestry of variation upon which the gentle hand of natural selection paints the slow, unfolding story of evolution. And with that understanding of life's incredible ability to subtly shift and adapt, we can begin to consider the intricate ways in which these variations are not just random chance, but part of the grand tapestry that makes each individual unique. Let's next turn our thoughts to how these genetic variations travel through families, shaping not just who we are, but sometimes our health as well. These variations we've explored take on even deeper meaning when we consider how they pass from parent to child. We now arrive at perhaps one of the gene's most profound and personal stories. How these tiny instruction sets shape us, not just in our everyday selves, but also in the delicate dance of human health and the inheritance of traits, sometimes including certain conditions. Imagine, if you will, that each of us is a magnificent, intricate library. Within this library, thousands upon thousands of books are shelved, each containing the precise instructions for building and running our unique selves. These books are our chromosomes, neatly bound volumes of genetic material, and within each book, every single word, every sentence, is a gene, whispering its particular instruction. Now, when we consider what makes each of us marvelously distinct, we find that while many of us have the book for eye color, for instance, the specific chapter might offer a subtle variation, perhaps for eyes the color of a summer sky, or rich hazel like polished wood, or deep brown like fertile earth. These variations in the instructions for a single gene are what scientists call alleles. They're like different versions of the same recipe in a cookbook. One recipe might call for a pinch of cinnamon, another for a dash of nutmeg. Both are delicious. Both are variations on a theme. Similarly, we inherit two copies of almost every gene, one from our mother and one from our father. It's as if each parent contributes half of their own unique library, and together they assemble a brand new collection for you. This delicate shuffling and combination of alleles is why family resemblances bloom across generations. And yet, each individual remains wonderfully unique. Now, sometimes, within these inherited blueprints, certain alleles can carry instructions that lead to a different path, perhaps one that might make our intricate biological machinery work a little differently. This is where the story of disease inheritance begins. For some conditions, the pattern is surprisingly straightforward, following rules first observed by a humble monk named Gregor Mendel, who studied peas in his garden. These are often called Mendelian inheritance patterns. Consider the concept of a dominant instruction. Imagine you have two architects, each giving you a design element. If one architect has a very loud, clear voice and insists on a particular feature, say a grand stained glass window, even if the other architect suggests something different, the stained glass window will likely be built. In human genetics, a dominant allele works similarly. If you inherit just one copy of a dominant allele for a particular condition, that single instruction is powerful enough to express the trait. Conditions like Huntington's disease follow this pattern. If a parent carries one copy of the gene for Huntington's, there's a 50% chance with each child that they will also inherit that dominant allele and potentially develop the condition later in life. It's a clear, unmistakable voice in the genetic choir. But then, there are the recessive instructions, like a quiet whisper. For a recessive allele to express its particular trait or condition, you need to inherit two copies of it, one from each parent. If you only receive one copy, the loud or normal allele from your other parent usually masks its effect. In this scenario, you would be a mycarrier. You carry the instruction, but it doesn't manifest in your own health. It's like having a secret recipe ingredient that only makes a difference if you double the amount. Conditions like cystic fibrosis, sickle cell anemia, or albinism often follow this recessive pattern. Many people are silent carriers of various recessive alleles, completely unaware until they have children with another carrier, and then there's a 25% chance with each child that they will inherit both recessive copies and express the condition. And the plot thickens further when we consider the very special sex chromosomes, the X and Y. Most of our other chromosomes are called autosomes, but the X and Y determine our biological sex, and they carry their own unique set of instructions. Conditions linked to genes on the X chromosome, known as X-linked inheritance, often show different patterns in males and females. Because males only have one X chromosome, paired with a Y, if that single X carries a recessive allele for a condition like color blindness or hemophilia, it will often be expressed. Females with their two X chromosomes have a backup copy. If one X carries the recessive allele, the other X can often compensate, making them carriers rather than affected. It's a subtle but significant difference in how the genetic story unfolds. But not all genetic stories are so simple, like a single clear instruction. Many common conditions, such as heart disease, diabetes, asthma, and even certain forms of cancer, are far more complex. These are known as multifactorial or complex disorders. They're not the result of one single gene whispering an instruction, but rather a grand symphony where many different genes play their part, each contributing a tiny note, sometimes amplifying or dampening the effect of others. And on top of this genetic orchestra, the environment plays a crucial role. Our diet, our lifestyle, our exposures throughout life. It's like baking a complex cake. The recipe has many ingredients, and the oven temperature, the humidity, and even the skill of the baker all influence the final outcome. Sometimes, the blueprint itself can have larger-scale typos or scrambles, not just a single word changed, but an entire page, or even a whole chapter either missing, duplicated, or rearranged. These are chromosomal disorders, like Down syndrome, which results from an extra copy of chromosome 21. These are broader strokes in the genetic canvas, impacting development in more widespread ways. It's an awe-inspiring tapestry, isn't it? Each thread a gene, each pattern a trait, weaving together the unique masterpiece that is you. The understanding of human genetics and disease inheritance is a journey of discovery, helping us to unravel the intricate language of life, offering insights and empowering us to understand ourselves and our families better. It is a testament to the incredible precision and delicate balance within each and every cell. Having seen how genetic variations can shape health across generations, we now arrive at a remarkable frontier, one where we're learning not just to read these inherited instructions, but to gently correct them when they've gone astray. We've spoken about the marvelous blueprint of life, the intricate instruction manual called DNA, and how its tiny segments, our genes, choreograph the symphony of our existence. Each gene is a precisely worded instruction, guiding the creation of proteins that form our cells, tissues, and everything that makes us, us. But imagine for a moment this incredible vast library inside each of us, filled with countless volumes of genetic information. Sometimes, though, within these incredibly detailed instructions, a tiny error might appear. A single word misspelled, a sentence accidentally omitted, or perhaps a whole paragraph out of place. These subtle changes, which we call mutations, can sometimes disrupt the harmony, leading to conditions that cause suffering and illness. For a very long time, these typos in our biological storybook seemed immutable, beyond our ability to correct. But then, science made a profound leap, discovering a tool of breathtaking precision, often simply called CRISPR. It's a name that might sound a little complex, but the story behind it is one of nature's ingenious solutions, harnessed by human curiosity. CRISPR, which stands for Clustered, Regularly Interspaced Short Palindromic Repeats, was first observed as a part of the immune system in bacteria. These tiny, single-celled organisms use it as a defense mechanism, a kind of molecular memory bank, to recognize and chop up the DNA of invading viruses. Scientists realized that this sophisticated natural system could be adapted, refined, and directed to perform targeted edits within human DNA. Think of our DNA, that winding double helix, as an incredibly long, detailed story written in an alphabet of just 4 letters: A, T, C, G. A specific gene, then, is like a particular chapter, or even a single important sentence within that vast story. Now imagine a tiny, incredibly clever librarian. Let's call it Cas9, which is a special type of protein. This Cas9 librarian doesn't wander aimlessly. It carries a very special map, a guide RNA. This guide RNA is like a unique address, precisely tailored to lead Cas9 to one specific spot, one particular word or phrase within the entire long story of the DNA. Once Cas9 finds that exact spot, guided by its RNA map, it acts like a pair of microscopically precise scissors. It makes a clean, accurate cut right at the designated location in the DNA. This creates what scientists call a double-strand break. Now here's where the magic truly unfolds. Our cells, these incredible tiny builders, have their own natural repair crews. When they find a break in their precious DNA story, they immediately set about fixing it. Sometimes, if we simply want to silence or disrupt a problematic gene, the cell's repair mechanism will patch up the break, often introducing small errors that effectively turn off the faulty instruction. In other times, and this is the truly revolutionary part, we can provide the cell's repair machinery with a brand new correct piece of story, a template, to use as they mend the break. It's like carefully replacing a misspelled word with the correct one, or inserting an entirely new beneficial sentence into the book of life. This ability to precisely snip and mend, to rewrite a specific genetic instruction, is what makes gene editing such a profound and hopeful advancement in medicine. Why would we want to perform this delicate molecular mending? The answer lies in the immense hope it offers to those affected by what we call genetic diseases. Consider conditions like sickle cell anemia, where a single letter error in the gene responsible for making hemoglobin causes red blood cells to become stiff and crescent-shaped. These misshapen cells can block tiny blood vessels, leading to excruciating pain, organ damage, and a shortened lifespan. With gene editing, scientists are exploring ways to correct that single error in the stem cells that produce blood, helping the body produce healthy, round blood cells once more, effectively rewriting the blueprint for a healthier blood system. Nor think of cystic fibrosis, a condition where a faulty gene leads to the production of thick, sticky mucus, especially in the lungs and digestive system, making breathing and digestion incredibly difficult. Imagine the possibility of gently correcting the instruction manual within the lung cells, allowing them to produce the correct protein and clear the vital pathways for air and nutrients. The potential is immense, moving us closer to treating the root cause of these diseases rather than just managing their symptoms. Gene editing's reach extends even further, touching on complex challenges like certain cancers or persistent viral infections such as HIV. For specific cancers, gene editing can be used to supercharge a patient's own immune cells, turning them into highly effective warriors armed with precise instructions to seek out and destroy cancer cells. This revolutionary approach is already showing incredible promise in therapies where a patient's T cells are modified to recognize and attack tumor cells. For HIV, it offers the potential to disable the virus's ability to replicate within human cells, or even, in ambitious future scenarios, to remove its genetic material entirely from the DNA of infected cells, offering a path towards a cure. This molecular mending can happen in different ways. Sometimes doctors might take specific cells, such as blood stem cells from a patient, gently edit them in a special lab environment, ensuring the corrections are made accurately, and then carefully return these corrected cells to the patient's body. This approach is often called ex vivo editing, meaning outside the living body, and has been particularly promising for blood disorders. Other times, the hope is to deliver the CRISPR tools directly into the body, to the specific cells that need mending without having to remove them first. This is called in vivo editing within the living body, like sending a tiny targeted repair crew on a mission to correct cells in the eye, the liver, or other organs. Of course, such a powerful and precise tool comes with great responsibility and requires immense care and ethical consideration. Scientists are working tirelessly to ensure these tiny molecular scissors only cut exactly where they are intended, avoiding any off-target edits that might accidentally change other unintended parts of the DNA. And the questions it raises are profound. What diseases should we treat? How do we ensure it's safe and accessible to everyone? How do we approach editing germline cells, which could pass changes on to future generations? These are thoughtful conversations humanity is actively engaging in as we learn more about this remarkable capability and strive to use it wisely and compassionately. So, as you can see, the story of our genes is not just a story written in the past, but one that is still being written, and with wisdom and care, gently edited for a healthier, brighter future. From understanding the intricate dance of transcription and translation, we now find ourselves on the cusp of truly engaging with the very language of life itself. And if we can edit these instructions with such precision, might we also learn to compose entirely new ones? As this quiet understanding of genes settles around us like the calm after a gentle rain, we might feel a sense of wonder at nature's intricate designs. For countless eons, life on Earth has evolved, telling its stories through the elegant language of DNA. Each organism a unique narrative woven from the same four letters, A, T, C, G. We've learned to read these stories, to understand the ancient wisdom encoded within every cell. But what if we could do more than just read? What if we could become not just readers, but also gentle storytellers ourselves, thoughtfully crafting new chapters, new verses, or even entirely new sagas within the grand library of life? This beautiful and profound concept brings us to the fascinating frontier of synthetic biology and gene design. Imagine, if you will, that the natural world is a colossal, ancient cathedral built over billions of years, with every arch, every stained glass window, every intricate carving representing a biological system. For a long time, scientists were like diligent historians, carefully studying the cathedral's existing architecture, trying to decipher the master builder's original blueprints. But synthetic biologists are different. They are the architects of tomorrow, the quiet, thoughtful engineers who, having learned from the existing masterpieces, now seek to design and build new wings, new chambers, or even entirely new structures inspired by the original principles, but with novel functions. At its heart, synthetic biology is about applying engineering principles to biology. It's about taking the building blocks of life, genes, proteins, metabolic pathways, and using them to design and construct new biological parts, devices, and systems, or to re-engineer existing ones for specific purposes. Think of it like a master craftsman who, after years of studying the art of clockmaking, begins to design entirely new clock mechanisms, perhaps a clock that tells time in a novel way, or one that not only tells time but also waters your garden. The materials are natural, but the arrangement, the design, is intentional and purpose-driven. And central to this endeavor is gene design. If DNA is the ultimate instruction manual for life, then gene design is the art and science of writing or rewriting those instructions. It's like learning the very alphabet of life, A, T, C, G, and then consciously arranging those letters into new words, new sentences, new paragraphs of genetic code. Instead of waiting for evolution to slowly, serendipitously stumble upon a useful new gene over millions of years, gene designers can meticulously plan and construct a DNA sequence to perform a desired function. They can be like gentle poets, carefully selecting each genetic word to evoke a specific biological outcome. How do they do this? The tools are becoming increasingly sophisticated, almost like delicate instruments of a cosmic scribe. One of the most fundamental is DNA synthesis, a process where scientists can literally print custom DNA sequences base by base in the lab. Imagine typing out a new blueprint on a special biological printer, creating a completely novel piece of genetic code from scratch. Perhaps a gene that has never existed in nature before, but which is designed to perform a specific, beneficial task. It's like composing a brand new musical score, note by note, ready to be played by the orchestra of a living cell. Then there's the elegant art of gene editing, often exemplified by revolutionary tools like CRISPR-Cas9. If DNA synthesis is like writing a brand new book, then CRISPR is like having a perfectly precise editor who can navigate directly to a specific word or sentence in an existing book, say, in the vast library of a cell's genome, and correct a typo, swap out a phrase for a better one, or even insert an entirely new sentence. This allows scientists to modify existing genes with exquisite precision, gently tweaking nature's own designs to enhance a function or fix a flaw. It's like a quiet whisper directly into the very essence of an organism's instructions. The potential applications of synthetic biology and gene design are as vast and varied as the natural world itself, yet they are imbued with a sense of thoughtful purpose. In medicine, synthetic biologists are designing microbes to produce life-saving drugs like insulin, or engineering immune cells to more effectively fight cancer. They're crafting entirely new genetic circuits within cells that can detect disease markers or deliver precise therapeutic interventions. Imagine tiny biological doctors designed to patrol our bodies, recognizing threats and healing with remarkable precision. In the realm of environmental stewardship, researchers are designing bacteria that can clean up oil spills by eating pollutants, or engineering algae to produce biofuels, offering a greener alternative to fossil fuels. They are working on creating biological systems that can capture carbon dioxide more efficiently, helping to restore balance to our planet's atmosphere. Think of these as nature's tiny, tireless custodians, but gently guided by human design to address specific challenges. Even in agriculture, gene design is opening doors to crops that are more resilient to drought, more resistant to pests, or more nutritious, reducing the need for harsh pesticides and making food production more sustainable. Imagine a wheat plant gently redesigned at the genetic level to thrive in challenging climates, providing nourishment to more people with less strain on the earth. This power to design life, to compose new biological symphonies, comes with a profound and beautiful responsibility. Like any powerful tool, it demands wisdom, foresight, and a deep respect for the intricate balance of the natural world. Synthetic biologists are not seeking to replace nature, but rather to learn from its profound wisdom and to collaborate with it to extend its incredible capabilities for the benefit of all life. It's about designing with humility, understanding that we are still merely apprentices in the grand workshop of creation, learning to wield our tools with care and purpose. As we consider the immense possibilities of writing new chapters in the book of life, we can also ponder the ethics and implications, ensuring that our designs are guided by a compass of compassion and a commitment to global well-being. This gentle journey into the realm of designing life's most fundamental instructions invites us to explore not just what is possible, but what is truly good and wise. And these questions of wisdom and responsibility bring us now to a new kind of landscape, one not just of adenine, guanine, cytosine, and thymine, but of quiet contemplation and profound questions. We've seen how these tiny, elegant instructions coiled within every cell orchestrate the very symphony of life. But what happens when we gain the power to not just read this symphony, but to subtly or even dramatically change its composition? Imagine, if you will, that your entire genetic code is a vast ancient library filled with countless volumes. Each book contains the story of you, your potential, your predispositions, the color of your eyes, the curl of your hair, the subtle hum of your internal workings. For centuries, we could only admire this library from afar, perhaps deciphering a few words here and there. But now, with our ever-advancing scientific tools, we've begun to learn how to open those books, to read the intricate tales within, and even sometimes to ponder, rewriting a sentence or two. This newfound ability, of course, comes with immense responsibility. It brings us to the very heart of the ethical and societal impacts of genetics. Let's think about genetic testing first. Picture it like holding a powerful, perhaps even overwhelming, magnifying glass. With it, we can peer into that library and discover specific passages about our health. For instance, prenatal genetic testing, like non-invasive prenatal testing, NIPT, or amniocentesis, can offer expectant parents insights into the likelihood of conditions like Down syndrome or cystic fibrosis in their developing child. Carrier screening can tell individuals if they carry a gene for a condition that could be passed on to their children, such as Tay-Sachs disease or sickle cell anemia, even if they themselves show no symptoms. And predictive genetic testing can reveal if an adult carries a gene that increases their risk for conditions like Huntington's disease or certain cancers, like those associated with the BRCA1 and BRCA2 genes. What do we do with such knowledge? It's a question that echoes with both hope and trepidation. For some, it offers precious time to prepare, to make informed reproductive choices or to proactively manage health risks. For others, it can usher in a tide of anxiety, ethical dilemmas about difficult decisions, or even discrimination. Imagine knowing years in advance that a serious illness might be looming. While empowering for some, this foresight could also cast a long shadow over their present, influencing life choices in unexpected ways. The very personal nature of this information raises profound questions about privacy. Our genetic code isn't just our story. It contains chapters shared with our parents, siblings, and children. Who has access to this deeply intimate data? How is it stored and protected? The fear of genetic discrimination, where insurers or employers might use genetic information against individuals, is a very real concern. Countries like the United States have attempted to address this with legislation like the Genetic Information Non-Discrimination Act, GINA. But such protections are not universal, nor do they cover every possible scenario. It's like trusting a precious, irreplaceable diary to a vast, interconnected network. We want to believe it's safe, but the potential for unintended exposure always lingers. Now, let's turn to an even more profound frontier: gene editing. If genetic testing is about reading the library, gene editing is about carefully, precisely correcting a typo or a misplaced sentence in one of those vital books. Technologies like CRISPR-Cas9, often described as molecular scissors, have revolutionized our ability to target and alter specific DNA sequences. The therapeutic potential here is truly breathtaking. Imagine correcting the faulty gene responsible for debilitating diseases like sickle cell anemia, cystic fibrosis, or Duchenne muscular dystrophy, effectively mending the broken instructions. This is called somatic gene editing, where changes are made in the body cells of an individual and are not passed on to future generations. But here we arrive at perhaps the most ethically charged crossroads: germline gene editing. This involves making changes to the genes in eggs, sperm, or early embryos. The crucial difference? These changes would be heritable. They would be passed down to all future generations, becoming a permanent part of humanity's genetic legacy. This opens the door to immense possibilities, but also to equally immense anxieties. On one hand, it could eradicate inherited diseases from entire family lines. On the other, it ignites the complex and often controversial discussion of designer babies. Where do we draw the line between healing a serious disease and enhancing traits that society deems desirable, like intelligence, athletic ability, or even physical appearance? Who decides what is a defect to be corrected and what is simply human variation? The very thought conjures images of a future where genetic advantages could exacerbate existing social inequalities, creating a deeper biological divide. This leads us naturally to the question of equity and access. These groundbreaking technologies, from advanced genetic testing to potentially life-altering gene therapies, are often incredibly complex and expensive. Will access be limited to those who can afford them, creating a genetic divide between the privileged and the underserved? If so, instead of benefiting all of humanity, These advancements could inadvertently worsen health disparities and deepen societal stratification. It's like building a magnificent bridge to a healthier future, but only allowing a select few to cross it. Moreover, as our understanding of genetics deepens, we are compelled to re-examine what we mean by normal, disease, or disability. Many genetic variations exist within the spectrum of human diversity. When does a variation become a condition to be fixed? There's a delicate balance to be struck between preventing suffering and celebrating the rich tapestry of human genetic differences. Historically, fears of eugenics, the discredited movement to improve the human race through selective breeding, serve as a stark reminder of the dangers of misusing genetic knowledge to impose societal ideals of perfection. At every step of this intricate dance between discovery and application, the principle of informed consent stands as a crucial guidepost. It means ensuring that individuals fully understand the nature, implications, and potential risks of any genetic test or intervention before making a decision. It requires clear communication, comprehensive genetic counseling, and a respect for individual autonomy in making such profoundly personal choices. Our journey into the ethical and societal impacts of genetics is not a stroll through a well-marked park. It's an exploration into a vast, often foggy landscape where there are no simple signposts, only complex questions. It demands careful reflection, open dialogue, and a deep commitment to compassion and justice for all. And with these thoughtful considerations settling gently in our minds, perhaps it's time now to rest, to let all we've learned find its peaceful place within us. And so, as the day gently fades and the world outside softens into twilight, we bring our quiet exploration of the gene to a peaceful close. We've journeyed together, not through vast cosmic distances, but into the profound, intricate universe nestled deep within each of us. It's a journey that doesn't demand grand adventures or loud proclamations, but rather a quiet wonder, a gentle curiosity for the unseen architects of life. Think of the gene now, not as a complex scientific term to be memorized, but as a whisper, a silent song playing deep within your very being. It's like a tiny, precious scroll, rolled up tightly, holding ancient instructions passed down through countless generations. Not a loud, booming command, but a soft, guiding presence, always there, always at work, even as you drift towards sleep. We imagined it tucked away, didn't we? Not in a faraway land, but within the smallest parts of you, those countless tiny cells that hum quietly with life. Picture them now, not as cold, sterile structures, but as miniature, wondrous worlds, each a silent library containing volumes of wisdom. And within these libraries, nestled amongst the long, spiraling staircases of life, are our genes. They rest there, patiently, serenely, like ancient guardians of your unique story, never rushing, always constant. These tiny instructions, these quiet architects, have been gently shaping you, guiding the unfolding of your very essence. They don't shout orders, they simply are. They orchestrate the gentle curve of your smile, the particular shade of your eyes, the unique rhythm of your laughter. They are the artists behind the softness of your skin, the strength in your bones, the intricate dance of every single cell that makes you uniquely, beautifully, undeniably you. And all this happens without a sound, without a fuss, a masterpiece being woven in perfect tranquility, moment by quiet moment. It's a thought that brings a deep, settling calm, isn't it? To know that within you lies such profound intelligence, such an ancient, living heritage. A silent thread connects you to every ancestor who walked before you. A shimmering line stretching back through time, weaving together stories, strengths, and even the quirks that make us wonderfully individual. And that same thread reaches forward, connecting you to the generations yet to come, a testament to life's continuous gentle flow, its enduring promise. It's a connection that doesn't demand action or effort, only a quiet acknowledgement, a humble appreciation for the miracle of continuity. As you lie here, comfortable and still, let these thoughts settle around you like a soft, warm blanket. There's no need to understand every intricate detail. No need to grasp every scientific explanation right now. The true wonder lies in the simple knowing, the quiet appreciation for the gentle, persistent hum of life itself. The gene, in its quiet power, reminds us that even the smallest, most hidden things hold the greatest stories, the most profound meaning. Now, as the stars begin their silent twinkle in the vast, dark canvas above, and the moon casts its gentle glow, feel yourself sinking deeper into the soft embrace of sleep. Let the images of these tiny, powerful guides, these ancient scrolls, these quiet architects, drift through your mind like peaceful clouds across a twilight sky. The journey of the gene, the story of life, is never truly over. It continues its quiet, miraculous work, even as your conscious mind rests. And perhaps, in the soft tapestry of your dreams, these quiet instructions will continue their gentle dance, weaving new patterns, fostering new understanding, or simply reminding you of the profound, calm wonder of being alive. Rest now, knowing that within you and all around you, the universe is unfolding its gentle, continuous story, always. Sweet dreams, little scientist. Good night.